Hi everybody, this is Andy from Med School EU, and today we're going to continue talking about protein synthesis and reproduction and inheritance and how it occurs. So the last part of Central Dogma of Biology is the process of translation, and we're going to talk in great detail about that. And we're also going to discuss the pre-mRNA processing that occurs before the process of translation. So I briefly mentioned how the mRNA is being processed after it is synthesized. However, today I wanted to go over in detail, just so you know the structures and the significance of it and the role it plays in the process of translation. So first, as soon as the mRNA comes out of the RNA polymerase and DNA complex, it's going to have a 7-methyl guanosine attached to it. So it's going to have the guanosine, which will have another methyl group, and it's going to have a triple phosphate. So it's going to connect 5' to 5', and that's going to be the 5' end that is going to be processed, and is going to prevent exonucleases attaching to the mRNA and degrading it. So it's very important that we have this 5' end, otherwise the mRNA would never be translated, as the mRNA would simply be degraded by our exonucleases that float in the cytoplasm. So next, we also discussed how it is terminated, how the mRNA is terminated, and then there's going to be a poly A tail attached to the 3' end. So just going over it again, how this happens is that the Rho protein is going to, of course, come all the way up to the RNA polymerase, and it's going to cleave the mRNA. So this mRNA will be cleaved, and it's going to make the 3' end processing. So the 3' end will be processed with a poly A. So this is polyadenylation. Not sure if I spelled that right, but it's polyadenylation that is going to attach the adenine bases, maybe 100 to 200 adenine bases, to again prevent exonuclease activity of the nucleases that float in our cytoplasm. And the last part of processing, so once we have the 5' end, and we have the 3' end processed, we're going to get something called splicing. So splicing, we're going to have introns. So these blue parts are going to be introns. And these orange or the red ones will be exons. So what the process of splicing, so the splicing, and splicing is performed by spliceosome. I'm not sure if that's how you spell it, but it's spliceosome. And what they do is they create these loops with the introns, and then finally they're going to cut away the introns, and it's going to connect the exons together. And the significance of that is, of course, it cuts off many of the introns, which would be the mRNA. A lot of the mRNA sequence is going to be cut out, and only the exons will stay. So this is another level of gene expression. So it's very important that splicing occurs, as some of these introns unwanted sequences will be cut out, and they will not be part of the amino acids that will come out of the translation part. Next, let's discuss the eukaryotic and prokaryotic translation initiation, because it's going to vary quite dramatically. Because if you could recall that prokaryotes do not have a nucleus, and so their transcription and translation is going to occur simultaneously. So while transcription is still occurring, we're going to get translation to start. So some of the ribosomes are actually going to attach to the mRNA that is still being synthesized. It's still coming out of the RNA polymerase, and translation is going to begin while transcription is still happening. However, in eukaryotes, so this would be the eukaryotes, it is going to be completely separate. And why is that? Well, because eukaryotes have a nucleus. So inside the nucleus, we're going to get DNA to RNA. So we're going to get transcription. So inside here, we're going to get transcription. And transcription must be fully complete before the mRNA moves out of the nucleus into the cytoplasm, where our translation is going to occur. So these processes are going to be separated in eukaryotes. However, they will be together with along in prokaryotes. Now, another way that eukaryotes and prokaryotes differ in terms of their translation initiation is how they do the reading. So the ribosomes in prokaryotes, so this would be the prokaryotes, and this one would be the eukaryotes. So in prokaryotes, the ribosomes are going to bind to a specific sequence called the Shine-Dalgarno sequence. So this is Shine-Dalgarno sequence, and this Shine-Dalgarno sequence is going to be several nucleotides upstream, so upstream meaning this way, of the AUG start codon. So AUG is the codon for the starting amino acid, which is methionine. So the tRNA that's going to come along will have the anticodon to AUG, which will, the tRNA will also contain the methionine amino acid, and that's how it begins. Now, in eukaryotes, we also have the AUG start codon, where methionine will bind. However, the difference is how it's read. So again, prokaryotes are going to have the ribosomes bind to the Shine-Dalgarno sequence. It's just a specific sequence of nucleotides that they're going to bind to, and then they're going to scan 5' to 3' to find the first AUG start codon, which will, from there on, it will begin its translation. However, in eukaryotes, things are a little bit different. What they do is that they bind to the 5' cap, so that's where the binding occurs for the ribosomes. And the ribosomes are going to have already a methionine attached to, or the methionine tRNA attached to the ribosome, and is going to scan in the 5' to 3' direction until it finds something called a Kozak consensus sequence. And notice that it had to skip several start codons in order to get to this Kozak consensus sequence, because not all AUG start codons are the start codons for eukaryotes. Only the specific one that resides in this Kozak consensus sequence, that is where the methionine tRNA is going to begin, and the ribosome is going to begin its translation from this AUG and onwards. But before that, it skips this AUG, it skips this AUG, because it's not within the Kozak consensus sequence. So that is the difference in the way they read their frame, the open reading frame, and the way they do the scanning in order to begin their translation initiation. So now let's look at some of the nitty-gritty details of translation elongation and how it actually occurs. So first of all, we need to know that ribosome has two units. It's got the small ribosomal subunit and it's got the large ribosomal subunit. The small ribosomal subunit is the one that binds to the 5' cap, and it scans in this area until it finds its start AUG codon, which is the one right here. And of course, it's already going to have the initiator tRNA, which has methionine. As you can see here, it has the methionine. And it's going to scan. Oh, great, it finally found the consensus sequence, the Kozak consensus sequence, where it has the AUG start codon, and it's going to begin its translation. So what happens next? How is it elongated, and what really occurs? First of all, we must recognize that the large ribosomal subunit has 3 different sites. It's got the E site, so that's the exit site. It's got the P site, which is the one that where the tRNA resides at this point. So I'm going to label it here. It's the T site. And of course, we got the A site. A site is called the amino acyl site. The P site is called the peptidyl site, and the E is the exit site. So that's very important to note. Now, another thing I must point out is this little enzyme, and this enzyme is called peptidyl transferase. It's going to have a significance in these later stages. So it's called peptidyl transferase. Peptidyl transferase. First, we must recognize that the methionine tRNA will be in the P site when it binds to the start codon. And that is where the amino acid is going to be attached. Now, moving along, the ribosome gets another sequence, the next codon, in the A site, and that's the amino acyl site. So that is where the next amino acid comes in and it binds. As you can see, that's the next tRNA where it comes in, and it binds to the A site. It binds to the amino acyl site. And in this A site, once it binds, it's going to have the complementary pairing between the codon and the anticodon of it. And what happens is the peptidyl transferase is going to cleave the peptide bond on the T site tRNA, and it's going to translocate it to the A site tRNA. Then we're going to use a GTP in order to translocate the entire ribosome. So the entire ribosome will shift over to the right side, and now the AUG start codon, so as you can see, the original tRNA that is empty, it's in the E site, it's going to be released. So over here, empty tRNA is released. And now we have the A site cleared. And of course, we have the amino acid chain growing on this tRNA. Now, of course, the next one is going to come into the A site, and the same thing will happen. The peptidyl transferase is going to cleave this and move it over, translocate it to the A site. And then we're going to use a GTP in order to shift and move over to the right again. And that will make the peptidyl tRNA empty because the amino acids have been transferred over to the next one. And it's going to leave, and the cycle begins all over again. So what's important to know is the significance of peptidyl transferase in translation. Another important thing is to know the 3 sites that exist in the ribosome, and knowing that ribosome is part of 2 subunits, the small and the large subunit. Peptidyl transferase is in the large subunit. Another important point is that the starting anticodon and the starting tRNA is going to reside in the P site. That is how it's going to begin, and it's going to elongate in this sequence as described. Now, another important concept that we must tie together with translation and how it occurs in the endoplasmic reticulum. It's very important, this concept, because it does come up a lot on the exam as well, is knowing how endoplasmic reticulum becomes rough endoplasmic reticulum. Because normally endoplasmic reticulum is just smooth. It does not have ribosomes. Ribosomes do not simply sit on endoplasmic reticulum and wait for the mRNA to come along and start its transcription. No. The ribosomes are going to be free ribosomes. So these are free ribosomes in the cytosol, so in the cytoplasm. So once the mRNA is released from the nucleus into the cytoplasm, these free ribosomes are going to attach to it, and they're going to start doing its translation and releasing its amino acid sequence. However, what it does is that as soon as the amino acid begins to come out over here, right, it begins to stick out like that, the top sequence, the N prime sequence, is going to have a signal peptide. It's called a signal peptide. And the signal recognition particle that also floats in the cytosol is going to bind to the signal peptide, and it's going to guide it towards the smooth endoplasmic reticulum. So this is the ER, but at this point it is smooth. It doesn't have any ribosomes attached to it. However, now that translation is occurring, this SRP is going to bind to the SRP receptor on the ER, and it's going to make it rough ER. And why is it rough ER now? Because it's got ribosomes attached to it. And ribosomes do not simply sit there. So if you were taught this in high school, that, you know, some of the endoplasmic reticulum is smooth and some of it is rough, and that's just the way it is. No, it's constantly changing. It's constantly changing. So sometimes it would be smooth if you're producing more lipid. Remember that smooth ER is going to produce lipids, and the rough ER is going to produce proteins. So of course, if you're doing protein synthesis, if you're doing translation, you're going to get more rough ER. However, if the cell does not need any proteins, it's not doing translation, then most of the ER is going to be smooth. So it's constantly changing between smooth, and it's constantly changing between the rough ER, depending on the demands of the cell. So, of course, once it's within the endoplasmic reticulum, the amino acid sequence is going to be released inside the lumen, and it's going to attach to the signal peptidase, which is another enzyme that is capable of cleaving amino acids. And, of course, once the amino acid has been completely formed, the signal peptidase is going to cleave it. So right here, a cleavage occurs, and it's going to be released into the ER. Now inside the lumen, it's going to do processing, which will fold the amino acid into its secondary and tertiary structures and quaternary structures, which will then be released and will be used within the cell. And finally, we're going to discuss translation termination. So translation termination, as you can see here, there is no stop codon, no stop anti-codon tRNA. So remember this, there is no stop anticodon. So we do not have a tRNA that has some sort of amino acid that corresponds to the stop codon. What happens is we have an enzyme called release factor. Release factor that will bind to the A site. So it's going to bind right here to the A site. The RF will bind there, and it's going to cause it's going to cleave right here. It's going to cleave the amino acids that will be released into the endoplasmic reticulum lumen. So that's the significance of it. RF is going to bind to the A site, which will cleave the amino acid from the P site and will be released. And then once that is cleaved, the ribosome large subunit will be removed, the ribosome small subunit will be removed, and everything is going to dissociate. And of course, our translation is now complete. Now, this concludes our lecture on protein synthesis. We know how the central dogma of biology works. In the next video, we are going to discuss prokaryotic DNA and the eukaryotic chromosome structure.